Good morning, everyone. At first, I would like to thank to the organizers for providing me the opportunity to talk in this conference. I am Rashidul Islam. Currently, I am doing PhD in bioinformatics at the University of British Columbia. So the project I will be presenting today is mostly done by a group of students who are studying different universities in Bangladesh. So where I am supervising them and leading this project. So in this project, we are comparing different assembly softwares for the de novo assembly of SARS-CoV-2 genome. Let's look into the genome of SARS-CoV-2. We know that this genome is 30 kilobase pair long. It has several genes. These genes are shown in different colors here. These genes are transcribed into mRNA and translated into different proteins that constitutes the major parts of the SARS-CoV-2. So the first genome was sequenced from Wuhan isolates from China. Since then, there are more than 50,000 genomes have been sequenced across the world. So this sequencing of genomes is helping us to understand the changes in the genome of this virus. That is allowing us to characterize the evolutionary trajectory of this virus in different time points and different geographical locations. So there are different sequencing technologies and there are different softwares that can be used to analyze those sequencing data. So today we will focus on the short-read sequencing technology. Using this technology, the entire genome of SARS-CoV-2 is chopped into smaller fragments. For each of the fragment, using this paired-end sequencing technology, we can sequence both the termini, left side is read 1 and right side is the read 2. So we will be able to know the DNA sequence of both the terminus, but not the entire fragment. And we can generate an entire genome looking into the overlap of those sequences. So this process is called assembly. So we use different software tools for the assembly of the genome from using these short reads. This is called de novo assembly. So there are several tools that can perform de novo assembly, but each of them have different assumptions how they will generate the contiguous sequence from the overlap. Also, they have different error correction methods, as well as they have different optimization parameters. So here we ask the questions whether one method is performing better than the others, or what is the degree of variability among different software tools for a given number of samples. So to study those variability, we downloaded the data from NCBI. The sequencing data submitted to NCBI has different assay types: amplicon sequencing, RNA sequencing, targeted capture sequencing, whole genome amplification, and there is category also for other. And we can see till June 2020, there are more than 6,000 samples have been submitted to NCBI. From each of the assay types, we randomly selected 100 samples, and we can see some of the samples failed in our quality control step, and these are the final number of samples we used in all our subsequent analysis. Here I am showing the list of assembly software tools we used. Some of the tools are well known for assembling larger genome, and some tools are popular for assembling smaller genome, and also some tools are used for metagenomic assembly. So there are several quality matrices that are used to find the quality of an assembly. So for example, one very basic approach is to measure what fraction of the genome have been assembled by a particular assembler. For example, this is the whole genome of coronavirus. You can see for assembler 1, we're able to assemble 50% of the genome. For assembler 2, 75%, assembler 3, 25%. But there is a fourth assembler that you are able to assemble the entire genome for the same sample. So we can clearly see the distinction of the quality for constructing the fraction of genome, which were assembler 4 performed better than the others. So here we are showing the fraction of genome recovered by different assemblers for all 416 samples we analyzed. We can see there is a higher degree of variability of recovery of the genome depending on the assembler. Looking into all of them, we can see that some of the assemblers were able to recover the 100% of the genome, almost 100% for majority of the samples, and some were only assembling 25% of the genome. So this is important to know, not all the assemblers are performing good for this particular data set. So here I highlighted red for megahit. We observed that not only for genome fraction but also other quality matrices, most of the times MetaHit is performing better for this Illumina paired in sequencing data to assemble the coronavirus genome. We have also looked at the standard quality matrices that are used to evaluate the assembly quality. For the sake of time, I will not show all of my data, but we are submitting our paper end of this month, hopefully. But I will show another analysis we have done. We have found some of the samples always have a better assembly independent of assembly tool, and some of the samples have very bad quality assembly whichever tool we use. For example, here I am showing an example of good quality sample that we performed assembly using the 4 different assembly tools, and we can see this is the genome coronavirus genome. The gray color is indicating the contiguous assembly, that means this region is assembled, where the red is indicating the gap in the assembly. This region was not assembled. So here in all 4 softwares, they were able to assemble the 99.99% of the genome. Here I am showing the example of bad assembly, where we can see most of the regions are red, where red means the gaps. These regions are not assembled. So even we use all 4 tools, none of them were able to construct the entire genome. We investigated more into it, and we showed that for the better assembly, the assembly quality of sequencing was much better. It has uniform read distribution across the genome, whereas we got bad assembly because the sparsity of the sequencing. You can see some of the regions have no read, so the assemblers were not able to construct the genome from those locations, whereas this region have some reads map, so some of the assemblers were able to construct the sequence from this region. So here the important point is not only the choice of software tools, but also the quality of the sample and quality of the sequencing also matters to get a good quality assembly. So this is my final slide. I want to acknowledge all the people who contributed to this project. So here we are working as a group where I am mentoring online. I'm teaching them and sharing our knowledge and on bioinformatics and doing projects on genomics. So if anyone is interested to learn genomics and bioinformatics, you are welcome to contact us and join our team. Thanks everyone for your time.